because why is the glycosidic bond in sucrose named alpha and beta? The reason why is, this took me forever to find because I was pulling out research articles, but so, and you guys will not need to know this detail for the exam. Let me say that. Just because it was after class. So basically, the nomenclature of the bonds for disaccharide molecules is based on the anomeric carbon. Anomeric carbon is the carbon, remember I talked about the functional groups, the C double bond O is the functional group for both aldehydes and ketones, right? You remember that from Monday. So that carbonyl group here is what forms the bond. When the structure cyclizes, this is the carbon in which the carbonyl group converts into this carbon. And so this carbon and all of the other disaccharide molecules, there's only one carbonyl group that makes up the bond. So that's why you only have, like, for the lactose is beta 1,4. But with sucrose, this sucrose is a keto sugar, and glucose is a aldehyde. And so just so happens when they cyclize that both of the anomeric carbons from each of the molecule, the sugar molecules, participate in the bond. So that's why this alpha comes from this alpha glucose, the beta comes from the beta fructose, and then the 1, 2, this is the first carbon, and then the 2 is coming from the second carbon. Does that make sense? Any questions about this? Again, this will not be on the exam. This level, so you do not see anomeric carbon.
I have a question.
Yes.
So in case of lactose, how do you know if it's going to be beta or alpha?
Lactose?
Yeah, because in lactose it's beta 1,4 with glycosidic bond, right? So how do you know could it be alpha 1,4?
No, because remember for well, I'll come back to that question. I have a slide for that. I saw another question in the back.
Yes, sir, I had a similar.
Okay, I'll come back to that when we do another question and see if this answers your question. So, any questions about sucrose? Okay. So, can't remember who, but someone asked how to generate the Hayward structures from the Fisher projections. This will not be on the test either, but here are the Fisher projections for glucose. Glucose is a cyclical Hayward structure, Fisher projection for fructose. So remember, this gives since the hydroxyl group is on the right side for both, that is the D isomer, right? That makes sense. So what happens is so if you look at the arrows are just telling you, the direction of the arrows are just telling you, when these molecules cyclize, the atoms on this side will be projected up, and the atoms on the right side will be projected down in the Hayward structure. So, in other words, if you wanted to identify the D isomer, we know the alpha and beta, what the alpha and beta isomers look like in the Hayward structure, because that's what we talked about. But if you want to identify the D and L isomer, basically this ends up being the 5th carbon, this carbon here. And so you would look at how this group sits. So because it sits up, it is the D isomer. But if it was reversed, it would be the L isomer.
So if the CH2OH was pointed down.
Correct. It would be the L isomer. Any questions? Okay. And someone asked about fructose being the only ketohexose. Yes and no. There are other ketohexose sugars, but they are not naturally occurring, and they're not like in biochemical pathways. They are generated in a lab, and they're used for like diabetes for no calorie sugars, or they're used in the industry for other uses. So D-fructose is the only ketohexose sugar that we study. Okay, now I'm gonna go to asking you guys some questions, and I'm gonna get to the question asked about the beta-1,4 lactose. So, first question: why is stereochemistry important? Anybody, one person, want to tell me why? Yes.
I believe from last time we talked about like thalidomide. You can have the same structural formula but a different spatial arrangement of one, and one could be for something like thalidomide was one of them. I think the L isomer or the D isomer was for
The RNS for there's so many
RNS. So
RNS.
the R1 was sedation, and the other one turned people into fish.
Yeah, correct.
Not good.
Great job. That's actually the example that I put for the answer. There are a couple of different answers, but great. Yep. So it does the flippers. Yeah, absolutely. So, any questions about that? What about what makes a molecule chiral? Yes.
4 different atoms on whichever carbon.
Correct. And I have that hint here. So 4 different single carbon bonds, and the 2 hints, of course, are below. You see the 4 different molecules. And as your classmate captured my heart with saying, it makes food taste good because it does. So, any questions about chirality? Yes, no? And then the last question. What saccharide is formed by beta-1,4 glycosidic? Lactose, is that the only one? Cellulose. Okay, so lactose and cellulose both have beta-1,4 glycosidic bonds. However, we as humans cannot digest cellulose. We do not have the enzyme. And so it was asked, why is the bond beta-1,4? And it's because since this is a beta isomer of galactose, right? The OH is on the top, right? That's beta isomer. And so this beta isomer is the only anomeric carbon in the bond. So when this cyclized, this carbon came from the carbonyl group, and so because this glucose, this other glucose molecule, this would be the anomeric carbon, but since you only have one saccharide that has an anomeric carbon forming a bond, forming the glycosidic bond, that's why there's only the beta-1,4. Does that answer your question? Oh, yes.
So with that then, so galactose is first in this presentation. Is there ever a time when the glucose and alpha isomer would be in front, which would then make it appear different? Or you're always going to see galactose
Yes.
first, indicating that's the beta isomer? Okay.
All right, now we're going to get into today's lecture, which is amino acids and proteins. Here are the learning objectives. So, with all the lectures, I'm going to try and just give you guys a foundation of why what we're studying is important. In the first block, it's going to be typically to understand how the foundational molecules work, whatever they are. And so, with proteins, we know that they're important in diseases. Their proteins are altered in disease states. Biochemistry loves to use phenylketonuria as an example. So we're gonna talk about phenylketonuria a couple of different times throughout this course. And in this condition, one of the enzymes, which is a protein, it doesn't work. So that's why we get this condition, and it leads to some toxic side effects. So understanding how proteins work, what they do, what they function as, and biochemical pathways is very important for us. And then drug metabolism, our body uses enzymes or proteins to metabolize like the majority. The 3A4 is, the cytochrome P450 enzymes are largely and heavily involved in the metabolism of drugs. So, and they are altered by different drugs, and they function different based on our genetic backgrounds in some cases. So, understanding how those proteins work in those situations is important as well. And then you guys will be diagnosing a lot of conditions, and you will use lab tests that are looking at protein levels in order to diagnose a condition and then therefore treat the patient. So ALT and AST is one of the major ones that looks at liver function. Those enzymes are found in the liver, and when that ratio is elevated, that means that there's liver damage. So you're gonna have to interpret those lab results and say, okay, this person has liver disease. What if he's on other drugs? What drugs can we give him that are less toxic to his system and things like that. So here again, I'm including these slides, the functional group. So another question I got yesterday was about knowing the structures of the molecules we talked about. So I'm gonna say, no, you guys do not need to know the structures of the molecules we talked about. You don't need to. I'm not gonna put up fructose and say, hey, tell me what sugar this is. I might say what functional groups are in fructose, and it's going to be the ones that we talked about, the ketone group or the hydroxyl group. Just in context of what we talked about and what functional groups are important, that's what you guys will need to know. Also, as we go along in the semester, you guys may need to know what components make up a molecule, and I'm going to get into that in the next lecture, but you will not be given a structure and say, oh, what structure is this? But you might need to know if a molecule has 3 different components that make up its base structure. You may see a question like that on the exam. So, functional groups that we're going to talk about in today's lecture are highlighted in yellow here, and so these functional groups, you will not need to know the amino acids that we're going to discuss today in terms of looking at the structure and identifying it. But the functional groups that we talk about today are very important because they're going to help explain how proteins form their secondary and tertiary structure, and we'll go into more details. So, how are proteins digested in our body? So, unlike sugars, protein digestion, there's no protein digestion in the mouth. It starts in the stomach and it finishes in the small intestines. And so what happens is the enzymes that break down proteins are in their inactive state. And so once we eat, those enzymes will be transported to their respective location. For example, pepsin is the active form of an enzyme in the stomach. It requires a low pH. So if its inactive form is pepsinogen. So but when pepsinogen is exposed to that acidic environment in the stomach, it's going to cause it to be activated. And then once it's activated, it will begin the process of breaking down proteins. And then protein digestion will finish up in the small intestine where several different proteins who require a high pH will be activated, and then they will break down those proteins into like either single amino acids or groups of amino acids that are then absorbed in the body via transport carriers, just like the sugar molecules are. So we're gonna talk about the specific protein, the specific enzymes and what they do in the later block, but for now, you just need to know generally what's going on, and that's how the digestion of proteins occur. And so we know that the amino acid sequence determines the protein's function. And so there are 20 amino acids that make up a protein. Proteins, as we know, are involved or are part of components of our hair, our skin, nails, cartilage, all things like that. And of course, as I mentioned, proteins are enzymes, and biochemistry is nothing but biological pathways, and so enzymes are a very important component of that. So, looking at the structure of single amino acid, there are a couple of components, and so here's an amino acid. Every amino acid will have an ammonium group, which is always going to be on the left. It's going to contain nitrogen atom, and it's going to be charged. It's going to contain, every amino acid is going to contain a carboxyl group, which will be negatively charged. It's always going to be on the right. And then it will contain a hydrogen group. All of these groups will be around central alpha carbon, and then the important group of the amino acid will be the functional group, and the functional group is going to let us know how amino acids interact with each other in biochemistry. We're going to go into more detail. So, because we have a positive charge group and a negative charge group on every amino acid, at physiological pH, which is the pH of our blood, it ranges between 7.35 and 7.45. At the physiological pH, an enzyme, amino acid that has a positive and negative charge, they will cancel each other out, and that is called zwitterium. Yes.
So the ammonia group is always going to be in position left.
The central alpha carbon. So the central alpha carbon is
Right. That's because it's balancing out the carboxyl group, right?
correct. So we're going to talk about different types or different groups of amino acids based on the property of them being hydrophobic or hydrophilic. So you have the non-polar, which is hydrophobic, doesn't really like water. And then you have the polar amino acids. And then we're going to break down the polar amino acids a little bit too. In that polar neutral, that means they have no charge. Polar acidic and polar basic. So if we were to look at, again, an amino acid structure, this is the amino acid glycine. The nomenclature of amino acids is based on, of course, their full name, and they have a 3-letter name, and then they also have a 1-letter name. So you guys won't need to know the 3-letter or 1-letter name because that's not important for you in terms of this course. But so we're going to just talk about the full name of each amino acid. So for the non-polar amino acid, these amino acids don't like water. So they're going to have functional groups that indicate that they don't like water. So those functional groups are aromatic rings, like you see here, or alkyl groups, hydrocarbon groups, alkyl groups are always going to have carbon and hydrogen atoms, and then a hydrogen group. So if you see hydrogen alkyl groups or aromatic carbons, those are always going to be non-polar, they don't like water. And so, because there are 20 different amino acids, when you look at these functional groups, sometimes they kind of are tricky, at least they work for me. And so, when I was in grad school, what I did was, and this trick has worked, it worked for me throughout grad school, but if I am not sure of an amino acid, what group they fall in, non-polar or polar, because they're a large structure, then I will typically look at within the functional group or the R group, I'll look at the first and last atom or molecule. And so, see that sulfur is kind of confusing. So what I have done is that's an alkyl group and this is an alkyl group. So that's how I know that this amino acid, methionine, is non-polar because it has alkyl groups. Does that make sense? Same thing with tryptophan. Even though I know aromatic rings are part of it, if I happen to forget that, during an exam, I look at this alkyl group and this hydrogen, and then I'm like, okay, yeah, those are definitely non-polar. Yes.
So is it the first and the last broken?
This is what I do. You guys don't have to do this, whatever works for you to remember this, but this is what I do. I look at the first and the last, and I'm gonna get into why in the next example, but is that clear? Does that make sense? Does that make sense? Okay. So those don't like water. Did you have a question?
Is that a good rule of thumb for each? Do you have like a good rule of thumb for each one? Or just for one?
Yes, this is what I've done, and it doesn't apply to every single amino acid, but this is what I've done, and it's helped me. So you guys, if it doesn't help you, then I understand, but if it does, then perfect. So polar amino acids, polar molecules like water. They have alcohol groups, they have that thiol group, which is sulfur and a hydrogen together, and they also have the amide group. The amide group is the C double bond O attached to a nitrogen and hydrogen molecule. So, again, same rule of thumb. See how there's an aromatic ring in tyrosine? If I looked at that quickly, I'd be like, oh, that's non-polar. But alcohols contain carbon, hydrogen, and hydroxyl groups, right? So see how this is carbon, hydrogen, and a hydroxyl group? Does that make sense? So this is like just a trick. Again, if it doesn't work, I'm sorry for bringing it up, but it's really helped me. So same thing. This is glutamine, and this is the amide bond. C double bond O N H. And so because in this one you see I didn't use the first and the last, but that amide bond I know what an amide bond looks like, so that keyed me into knowing that this is a polar, and because there's only one plus and one minus charge, that's why it's neutral in physiological conditions. Yes.
Is the sulfur hydrogen, that's the thiol?
Yeah, sorry, the sulfur hydrogen is the thiol. This one is very important for disulfide bonds. So we're going to talk about disulfide bonds. So this is kind of unique in the groups, but this one, remember there was a sulfur in this group, and it can't do anything because it's buried in between non-polar molecules. So, but the sulfur in this group is exposed, and so that's why it is polar and able to interact with other molecules. Yes.
So those aromatic rings, so that is another carbon ring?
It is.
Okay. So it's central carbon with
Yes.
not important what comprises those.
No. Yes.
That one is non-polar,
it is because of the aromatic ring. But again, you see that it had, I could have circled this too, it has that. Oh, sorry. Oh, sorry. It's non-polar because all of these are non-polar, but sometimes that aromatic ring like throws me off because it, you know, it appears in other, in the polar, some polar amino acids. So these, it just so happens that these three groups are all non-polar. But for me, like I said, I just look at the first and last. If I happen to forget that this is a non-polar, I'll look at the first and last groups, and I know for sure that it's non-polar because alkyl groups and hydrogen are non-polar. So polar acidic amino acids. So these, yes.
Sorry, just, can you go back?
No, sure.
So just to clarify, the first three on there are from left to right. The first three are alcohols. The thiol is the sulfur one. The last two are amides.
Yeah, sorry, yeah, I didn't specify. Yes, so CH2OH alcohol. So serine, threonine, and tyrosine are considered alcohol because they have that carbon, hydrogen, and hydroxyl group. Cysteine is the thiol, and then asparagine and glutamine are the amide bonds. So polar acidic amino acids. So I didn't say this, but this ammonium group is basic. It's when it has a nitrogen, that nitrogen typically is a basic molecule because it's a hydrogen acceptor, and carboxylate group is considered acidic because it's a hydrogen donor. And so in polar acidic molecules, they have an additional carboxylate group. You see the additional carboxylate group here for aspartate and glutamine. So under physiological conditions, these are gonna always be negative, right? Because you see these two cancel out and then you still are left with a negative charge. Is that clear? Polar basic, they have these amino acids, histidine, lysine, and arginine have an additional ammonium group, and that's going to be positively charged nitrogen and hydrogen molecule. That's why I circled those. And so these amino acids are going to always be positively charged under physiological conditions because they have that extra charge. These two cancel each other out, and you have an extra positive charge. Does that make sense?
I'm sorry.
Yes.
Could you explain it again for me?
So these are histidine, lysine, and arginine are considered polar basic amino acids because we know that nitrogen is basic because it's considered a hydrogen acceptor. So in the backbone that we talked about, here's one ammonium group, and here's the carboxylate group. So these groups will cancel each other out, opposing charges. But you're left with a positive charge for each of these. So under physiological pH, these amino acids will be positively charged. Okay. So proteins are formed by amino acids that are bonded together. And that makes up when amino acids are bonded together, that is the primary or structure of amino acid. It's simply the sequence of amino acids bonded together. And here's an example. We're going to talk about that in a little bit more detail. But the nomenclature of peptides, if it's 2 amino acids, it's a dipeptide, 3 tripeptide, and then if you have several amino acids bonded together, it's a polypeptide. You won't need to know the exact number that makes a polypeptide. Just typically, if there's only 2 right here, call it a dipeptide because there's 2. So the formation of peptides and amino acids, they're formed by amide bonds. So what happens is so remember, in an amino acid, the ammonium group is always going to be to your left, the carboxylate group is always going to be to your right, as shown here in glycine, alanine, and the ammonium group is in blue, the carboxylate group is in pink. So when these two amino acids combine, the carboxylate group from the first amino acid interacts with the ammonium group from the second. So what happens is we lose a water molecule. It's a dehydration reaction, just like we saw with the saccharides. And so you have the amide bond here. Remember, amide bond is C double bond O bonded to an NH. So that is an amide bond, and it's also called a peptide bond. So basically, a primary structure of amino acid is when amino acids form together through peptide bonds and or another name for a peptide bond is an amide bond. Does that make sense? So here we see an example of two amino acids, and if there were three amino acids, it's the same thing. So, four amino acids, the same thing. The carboxylate group of the first or prior amino acid will interact with the ammonium group from whichever the subsequent amino acid is. So this one has three amide bonds or three peptide bonds. Does that make sense? Okay, so here is just an example of protein structure. Insulin was the structure was discovered just only 100 years ago, which is interesting how fast that technology and research has, how fast we've advanced, because now there's a lot of drugs targeting, insulin and the pathways involved in insulin and what it's doing. And so in insulin, there are two polypeptide sequences formed together by what bond makes, what type of bond is this? Disulfide bond, two sulfide groups. So we see three different disulfide bonds with insulin. So this is polypeptide with two chains. You have A chain and B chain. This is just one example. What's interesting about insulin is that, prior to us understanding the structure, we knew some of the side effects that it caused and the conditions it caused in patients, and it was considered a very deadly disease. And so what was a recommendation by clinicians, you guys, which is still a recommendation if somebody has insulin issues, is to give them a diet that's low in carbs. Insulin, whether it works or not, we're going to talk about when it works or not in the types of diabetes. If we have a higher protein diet, then the insulin won't have to act on the carbohydrates, and the carbohydrate levels will be maintained more steadily. So there are 9 essential amino acids that we must acquire from our diet. They are histidine, isoleucine, leucine, lysine, thymine, phenylalanine, threonine, tryptophan, and valine. The other 11 we can make in our body. And so sometimes you might see asparagine as being an essential amino acid, but it's conditional. So if a person experiences trauma and is burnt, has severe or significant burns in the body, then asparagine becomes an essential amino acid, and asparagine is found in foods. And so this is an acronym. I don't know if you guys have seen this before. This is an acronym that helps us remember the 9 essential amino acids. private Tim Hall, sometimes with the A or without the A, but this one is only a conditional essential amino acid. So this is just a mnemonic or acronym that you use to help you remember the non-essential amino acids. Any questions? Have you guys seen this one before? This acronym? No? Yeah, we used to make up our own when I was in college before computers. Yes.
Not with regards to this group, but
Can you speak up? Sorry.
sorry, not with regards to this slide specifically. However, since the 11 amino acids that we can synthesize in the body, those are branched chain amino acids, correct?
The 11 amino acids. I don't know. They might be. Some of them I'm sure are, but I don't remember the exact classification of the 11. But I know that our body can make them, and we're going to talk about in block 2 how our body will make different amino acids. Any other questions? So, the primary structure of amino acids is the amino acids bonded together by peptide or amide bonds. Secondary structure comes into play, and those are formed typically by a hydrogen bond. And so, what happens is the secondary structures occurs when molecules from two amide bonds that are in close proximity interact together, and they form folding of the amino acid polypeptide chain. And so two examples are alpha helix and beta pleated sheath. And so in both of the examples, it's when the C double bond O of one amide group interacts with the NH of another amide group that's in proximity. So that they do that through a hydrogen bond. And so when the secondary structure is the alpha helix, the proximity of the amide bonds creates a helical-like structure when the hydrogen bond is formed. And the beta pleated sheet, again, same molecule, C double bond O interacts with the NH to form a hydrogen bond. But in this case, the proximity of the amide bonds creates like a folded pleated sheet pattern. And so what I've done is I've just kind of blown up the different bonds, hydrogen bonds with the alpha helix and beta pleated sheet. And so you see the C double bond O from one amide group interacts with the NH from another amide group, creating a hydrogen bond between the oxygen and hydrogen. And it's the same thing. The black molecule is carbon, the red is oxygen, the blue is nitrogen, and the white is hydrogen, again, creating a hydrogen bond. Does that make sense? So these structures, these alpha helices or beta pleated sheets, they can appear in protein by just that secondary structure, or you can have both in one polypeptide, and that's of course going to be based on the sequence of amino acids, because that's going to dictate how close the molecules are to each other based on the arrangement of the side groups. So another secondary structure that is created by hydrogen bonds, and this is a structure that gives strength and support to our collagen. It is a triple helix, and that's like a braided-like pattern where you have three polypeptide molecules in proximity, and they create hydrogen bonds, and essentially looks like a braid or a tightly twisted molecule. Does that make sense? Okay, so come to our first check on learning. So with the word bank below, identify the type of protein structure. What is the protein structure? Polypeptide chains held together side by side. Beta pleated sheet. What about the sequence of amino acids? Primary. Corkscrew. Alpha. And 3 peptides woven together like a rope or braid. Does that make sense? Easy peasy, right? Okay, but what, oh yes, I'm sorry.
Sorry, sir, I was just gonna ask, so like for us to be able to recognize this, will we be given either like the description of the peptide like that, or just the picture?
Yes.
Like this?
Yes. So there are 2 other structure types. We talked about primary and secondary. There are also quaternary and tertiary. Quaternary structures are, there are several different types of interactions that we're going to discuss in quaternary structures. And then, I'm sorry, in tertiary structures, there are several different interaction types that we're going to discuss. But in the quaternary structure, the general definition is that it consists of 2 or more polypeptide chains. And so I'm going to put you guys on a 10-minute break and then we'll pick this back up.
Yes. So we no, still on Friday. We don't have meat on Friday. We take one on Friday. We have him in the morning. The part lab for 2. Tomorrow's the last 2. So we only do twice the week after.
I started with Sylvia. It seems a lot of follow-up.
Yeah, so it's not even. So when we talk about this next topic, tertiary.
I just want to know.
Yeah, no, no, that's the one, sure.
So yeah, so
So let's say don't make yellow snow. Let's say maybe asparagus maybe depending on the day.
I'm curious,
Yeah.
you see how it gets like machine. I mean,
Rails and I was like, anything I lunch sucks. Even though for me, I actually need to like take anything and have to do a bunch more repetition. I just love it. Actually want to do. I think it'd be more like I already had a general thinking. I gotta wonder.
that's like basically.
No, listen.
Never go wrong doing it.
Wait, was that? What's up, man? How are you? I'm good. I'm like looking. He's explaining it today. I'm like, see, is it like to me? Like, I think it makes more sense than buying cars. Cars, I'm like, I gotta go through it. Yeah. And I was just telling them, I was on the side. Easier just for me to like look at it and identify it. See it. I can tell like, this is like my first time looking at all of these things. I feel like trying to puzzle something that different people have different
Basically have to turn around here and start studying. I mean, it helps. I already have it all planned out.
That's exactly what
We're gonna continue on with tertiary structures. So I got a lot of great questions up here. Y'all are making me forget how much, y'all are making me realize how much stuff I forgot. That's okay. The answers for you, I kind of like doing that. It's keeping my old brain active. So one of the most important questions is, will you need to identify amino acids by site? No. And we're going to talk about how you may see a question on the exam. So that's why I want you to focus on the different functional groups. So the tertiary structures of amino acids have functional groups, and these tertiary structures are formed by different types of interactions based on the molecule's amino acid sequence and functional group. And so the first type of interaction we're going to talk about is hydrophobic interaction. So hydrophobic interaction would be formed by R groups that are hydrophobic. So it would have to come from two R groups. And what I know you just had this within this lecture, but what are the non-polar R groups that we discussed? Aromatic rings, what else?
Hydrogen.
CH alkyl groups,
Hydrogen.
hydrogens. So, not saying you will, but you may see a question on the exam. And you need to know the different functional groups. And you know these functional groups, you know they're all non-polar, so non-polar, hydrophobic. So what these groups are gonna do, they are going to, around proteins, typically there's aqueous environment. These groups are going to want to repel that, get away from the water, and so they're gonna form like a non-polar center, if you will, around like amongst themselves. And so of course that's gonna cause the protein sequence to fold and bend in different ways. Does that make sense? So, you also have hydrophilic. So we know hydrophilic, it has to be a polar amino acid, right? So one of the polar amino acids that we looked at, we didn't even name it, but you know that it's polar because it's an alcohol group, right? That's the C2OH, that's alcohol. So we know that's polar. So what this polar amino acid is going to do is cause a hydrophilic interaction with the water molecules that are surrounding the protein. So they're going to want to move towards that water. So that is also going to cause the protein to fold and bend, and the R groups to fold and bend to attract, essentially it attracts the water. Okay. Another type of tertiary structure is salt bridges. Salt bridges are going to be formed when you have opposing charge amino acids. So you know that, and this is at physiological pH, so you know there's only two groups, and that would be the polar basic and polar acidic, right? So when there are opposing charges at physiological pH, you're going to have a salt bridge interaction, and so they're going to attract each other and also cause particular conformational 3D structure of the protein. Then hydrogen bonds. I'm kind of sick of talking about everything is a hydrogen bond. It seems like that's common, and so even with tertiary structures, we talked about different examples of hydrogen bonding in the secondary structure, but tertiary structure also have hydrogen bonding as well, and that's going to be when you have polar group of one amino acid interacting with an oxygen or nitrogen in another polar group. So you would just need to know the functional groups that are polar. And if you see two polar functional groups, then you're gonna it's a tertiary structure and it's a hydrogen bond. Does that make sense? Yes.
Would there ever be a polar group with one of the polar basic or acidic in this situation? Or is it
No.
just polar?
No, because that was like that. They might have some type of interaction to each other, but let me say there may be, but it's not gonna be any one of these interactions. It's not gonna form the type of interaction that we see with like the salt bridges from that, or with this,
Okay.
because the polar acidic, polar basic, they have a charge, so it's gonna do something a little bit different. And then we talked about the disulfide bond. So of course, that's always gonna be between two cysteine amino acids, right? Those thiol groups will interact. Does that make sense?
Disulfide is always between two systems.
Always gonna be two cysteines, always. So within one protein, you can have all of these different types of interactions, all five we talked about. And this is just one polypeptide sequence. And so if we want to go into quaternary structures, then that will be at least two polypeptide sequence amino acids forming all of the types of interactions that we just discussed. So same types of bond, but instead of within the polypeptide sequence, it's going to be enter between two or more different polypeptide sequences. So hemoglobin is an example of that. Hemoglobin has two alpha and two beta chains. You don't need to know anything about hemoglobin other than that it is a quaternary. It has quaternary structure because it has the four different polypeptide sequences, and hemoglobin has that heme group which has iron in that it's responsible for transporting oxygen. But you don't need to know any of that for this, but just hemoglobin is an example of a quaternary structure protein. So proteins can have primary through quaternary structures. And here are examples of each. You have the primary, secondary, alpha helix, tertiary, quaternary, two or more polypeptide sequences. So when we look at how proteins are affected, we know protein hydrolysis occurs when proteins are digested in our digestive tract, and that is done via the appropriate amino acids or proteins that we talked about at the beginning of the lecture in the digestive system. But protein denaturation, there's several different types of denaturation, and we're going to talk about the different types. But first, just going into a little bit more detail about hydrolysis. So protein hydrolysis breaks down the primary sequence of amino acids, meaning that it breaks down that peptide or amide bond, and it's when that bond is formed, it's a dehydration reaction. So of course, in order to break it, we're going to add water back to it. And so here's just an example of 3 or 4 amino acid sequence that has 2 bonds that are 2 peptide or amide bonds that are broken via water and digestive enzymes. And so that will give you 3 singular amino acids. So now denaturation is a little bit different. So denaturation disrupts the secondary, tertiary, and quaternary structures of proteins, and it renders the proteins inactive. So they're no longer active if they do not have the three-dimensional structures that are disrupted. And so here are some different examples or ways that protein denaturation can occur. So increased temperature, change in pH, mechanical agitation, and so if any of these happen, that's going to disrupt the interactions that we just talked about in the tertiary structures, and that's going to linearize the protein, and then of course, when the protein is linearized, it's no longer going to be active, biologically active. So heat, we know, like when we cook meat or we cook eggs, that the proteins break down to make the meat more tender, and in eggs, we notice that the egg change from clear to translucent, the egg white, and that is just the disruption of the hydrogen bonds and the interactions. And so, but clinically, if we get a high temperature, like above 105 degrees Fahrenheit, 110, that will cause proteins, particularly brain proteins, to denature, and that can lead to coma and even death because our brain proteins are no longer active, and we know that's very important. Which is why if we have fevers and stuff, they don't. The providers, you guys will not want our temperatures. I think the cutoff is like 104 degrees, and it becomes significantly more dangerous. But so that's a clinical way that protein denaturation will disrupt bonds. In terms of the food, the heat does not change the nutritional value, of course, but it will change the interactions. And so when our proteins are exposed to acid and bases, that's going to break the salt bonds between the polar acidic and polar basic functional groups. And so, for example, we know that, and we're going to talk about this in more detail, but there are digestive enzyme pepsin. It's in the stomach. The stomach pH is very low. Trypsin is the active form, active enzyme that breaks down proteins that's in the small intestine. So the pH for the stomach, the average is about 2, which is very acidic. The pH for the small intestines is 7, 8, and so those enzymes can only function in the respective environments that their sequence requires. And so if pepsin were to be floating around in the small intestine or trypsin in the stomach, those enzymes will not be active because they're going to denature because they're in a different environment. So all of the bonds, the interactions that we've talked about will be disrupted. And so alcohols or organic solvents or compounds also disrupt or denature our proteins. And they do this because in aqueous environments, water forms like a hydration cell around the protein. In this is a picture to your left, like a little schematic. What happens is, if you use hand sanitizer or whatever, say that's a bacterial protein on your hand, the hand sanitizer will interact with those water molecules and the proteins within the bacteria, so it's going to break those hydrogen bonds and interactions, and the protein will linearize, and that's going to kill the bacteria or make it inactive in terms of what harmful it can do if you need a sterile environment or things like that. Does that make sense? And then heavy metals. So heavy metals, lead, mercury, and silver, they displace the metals in our body, like for example, iron. So what happens is mercury and lead will displace iron, which is found in heme, as I just mentioned, and so it will irreversibly bind, and then like that will make heme, that will inhibit heme from being able to transport oxygen and do other functions that whatever enzyme is associated with it can do. And so we're going to talk about how to treat metal intoxication in block 2, but we know that it denatures proteins because it interacts directly with the groups within, like even if there's an endogenous metal ion within the protein, and it can also interact with the carboxylate group in the protein. So it's just gonna jack it up, and it's typically irreversible. So our body will need to regenerate more of that protein in order to like circumvent or counteract the effects of mercury or poison intoxication. And there are also some treatments too that we can use in the immediate response, but we'll talk about that in the next block. And then, so if we whip an egg, like mix it up to make scrambled eggs, that's disrupting the hydrogen interactions. Also, if we make a liver meringue pie, liver meringue, the meringue is made out of egg whites. That whipping will break the hydrogen bonds, and that is also an example of how mechanical agitation will break the bonds. So what it's doing, that whipping will stretch the bonds until they eventually break, and then that is disrupting the tertiary, secondary, or primary structures, depending on what type particular protein has. So this is a summary of what we just talked about in terms of denaturation. You may maybe see a question about this on the exam, so I would use this chart as a study template, maybe. If you want to, you don't have to. No. Any questions? Right, thank you. Thank you. Thank you. Any questions? All right, I will see you guys on